E agora? Conheço a cena em grande.
Tens que mudar aqui o som.
Deixa eu pôr o som por aqui.
O vírus.
Ah! Isto está ligado? Não está ligado.
O cruzinho não está ligado. Acho que desliga.
Good afternoon. You can hear me well? Yeah.
Yes. Yeah.
Yes, great.
Yes.
So welcome everyone to the second day of the webinar, Identification, Biology and Management of Insect Vectors of Xylella fastidiosa. This has been a two-day event that will provide an up-to-date overview of the plant pathogenic bacterium Xylella fastidiosa and its vectors. So this workshop, as you already know, also includes practical sessions focused on insect vectors and potential vectors. And it was supported by CITED, IberXIFAs, the Ibero-American Network for the Vigilance of the Xylella fastidiosa, and also by the International Olive Council. in the International Olive Council, our work would like to congratulate the Instituto Politécnico de Bragança for this initiative that is really important to combat and to prevent these so complicated problem. So unfortunately, I will not be able to be here with you today during the afternoon. So I will pass the floor to Isabel Rodrigues and hope that we may organize many of. I wish that we would not be needed, but I hope that we can organize other of these sessions real soon, because as stated at the IOC, we really think this is really important for the sector. So have a great work. Thank you so much for this opportunity. Thank you so much.
Thank you, Katarina, for the introduction. Now we're going to start the second day of this webinar. First, I would like to ask the participants to ask questions during the presentations, because you have a little delay between the Zoom and the YouTube. So please ask the questions during the presentations, not at the end of the presentations. So now we're going to start the second day. And our first invite speaker is Carlos Chacon. Dr. Carlos Chacon has a bachelor and a master in microbiology in the University of Costa Rica. He also has a PhD in microbiology at the same university. Currently, he is an associate professor at the University of Costa Rica, College of Microbiology of the Department of Bacteriology. His research focuses on bacteriology and microbial pathogenesis, with several publications related to Xylella fastidiosa in collaboration with different research centers. Today, he's going to tell us about the situation of Xylella fastidiosa in Central America region. Carlos, you can start.
Thank you, Isabel. I will project the, let's see. Can you see my screen?
Yes.
Thank you, Isabel, for your kind presentation. And I would, before I start, thank the organizers for this invitation. My talk for you today is about Xylella in the Central American region. I will try to give you insights into Xylella in our geographic environment, and especially I will focus on Costa Rica. That is what we work on. So the first thing is that Xylella fastidiosa in the Central American region has a big question mark. There's a lot of work and potential work to do in this region. There is really a lack of scientific information in the terms of biology of the pathosystem, but also in terms of epidemiology and other things that are important to understand and to take decisions about this pathogen. So I will talk about what we know and eventually try to give a scenario of the importance of studying Xylella fastidiosa in this region. For purposes for this presentation, I will include Mexico in some part of this presentation, but Mexico is really part of North America. So if we look at the Xylella situation, or this is what you get for Central America. You can see that we are right here in the center, and we are flanked by North America, where you can see all the spots of the presence of Xylella in the south part of the United States, and also in Mexico. And the Central American and the Caribbean islands are flanked between this really positive or where Xylella has been detected and has been a problem, an agricultural problem throughout the years, and South America, where the other region that is well known to have Xylella issues is Brazil. But as you can see, Venezuela has also official reports. In this case, the official reports for this Central American region, including Mexico for this purpose, would be Mexico, Costa Rica, that is what we're gonna talk about our situation. And there are official reports of Xylella in Puerto Rico. If we do a search for more information about the region, you can get some other publicly available reports, including this one I put here for Guatemala, where they have studied Xylella on coffee with negative results, but have found some positive results in ornamentals. This is an academic work for a university in Guatemala. In the case of Honduras, there has been reports of Xylella on coffee plants, especially from European interceptions from coffee plants that were exported and where Xylella was detected. And in Nicaragua, there are some studies in relation to citrus and vectors regarding Xylella fastidiosa. But as you can see, there is a lot to be done. There is all of these gaps to be fulfilled, since this is a region that we share a lot of our economic and biodiversity conditions. So the possibility that there is more diversity and potential of studying Xylella is a lot, is great. And there is a lot to fulfill in this field. Regarding Mexico, I will start with Mexico. They have reported Xylella fastidiosa, especially regarding vineyards. On the map on the left, you see all the zones, geographic zones, where grape vines are planted in Mexico. You see that it goes from the center of the country up to north, to the northern part, where in the border with the United States. And in this part, they report the presence of pear disease. So they have to deal with pear disease in these geographic latitudes that are very near or closer to what would you expect from California or the southern part of the United States. These persons, Aguilar, Granados, and collaborators did a study where they isolate Xylella from these coffee, from these grapevines. And well, they studied different grape vines in different parts of the country, and they isolated Xylella from some of these parts. As you can see, these are the red dots here in the northern part of Baja California and in Querétaro. When they do an MLST analysis, they see that the ST types they find are related to Xylella fastidiosa. This is more expected. And interestingly, the ones that are near the border with the United States are similar to California strains. This is expected, but it's interesting that the one down here in Querétaro is the ST type is more related to the ST types that have been reported in Costa Rica. This is important because since we have a gap of knowledge of the types of ST that are in the Central American region, at least we know that in the central part of Mexico, we can get some ST types of Xylella fastidiosa that are similar to the ones that are circulating or are present in Costa Rica. There have been other work done in Mexico. For example, this one is of mapping of potential host plants of Xylella according to the list of hosts. And with this mapping, what they try to do is like a risk map of places according to the presence of these plants, the possibility of finding eventually Xylella fastidiosa if something in the environment changes, and it would give some kind of outbreak. So this is kind of a risk map taking this information as a basis to have this like preventive exploration or diagram as a risk assessment for Xylella eventual outbreaks. Moving on, I will, as I told you, there is little information on the other countries in Central America. I will focus from now on what we know from Costa Rica. Something that is important is where our geographic and climatological conditions. Costa Rica is a really small country. Most of our population is right here in the center of the country, in a central valley. We are around 5 million people, and around 3.5 million people live in this circle you see here. Most of our territory is between 900 and 1800 meters above sea level. We have a big high rocky area in the country. We have two seasons: rainy season that is from May to November, and dry season from December to April. And we usually have very stable temperature, and the difference is between lowlands and the highlands, where the range of the temperature, although stable, is it has a kind of range. Here you see from 22 degrees to 37 in lowlands, and in highlands from 18 degrees to 32 degrees. But humidity levels usually and temperatures are very stable throughout the year. This is very important because maybe as we don't see some drastic changes in our conditions, this may favor that we don't see so many symptoms in relation to Xylella fastidiosa. We also have, as another important aspect related to Costa Rica, is our biodiversity conditions. We have more than 120,000 species known in the country, and we account for 6.5% of the global biodiversity. It's a little country, but as you can see, we have a lot of different types of tropical forest, from dry forest to very wet forests. And this gives all of these microclimates and micro conditions gives us in a small piece of land a lot of potential and diversity. And this also may contribute to what we see around Xylella. The other important aspect is related to crops. Costa Rica is known for growing pineapples, bananas, and coffee that is usually grown in the central part of the country in highlands. So the distribution of agricultural land that is really immersed in tropical forest is still very evident. And here in the right, you can see a picture of a coffee plantation and its surroundings. And if you move out, although it is dedicated to coffee, you will see a lot of tropical forest, secondary or primary forest surrounding these coffee plantations. And this also has an impact on the way that this pathosystem may impact or not these crops where we know that it is present. From a historical point of view, the first report of Xylella fastidiosa in Central America was done in Costa Rica in 1979. In this year, the bacteria was not known as Xylella fastidiosa, but the plants were grapes that some people from California brought some grapes. They wanted to breed and try to have grapevines plantations here in Costa Rica. And these grapes got Pierce disease symptoms. They took the plants back to California and they isolated the bacteria and everything, all the biological and serological experimentation they could do. This was a rickettsia-like bacteria that's how it was known in that time. And we know now that this is Xylella fastidiosa as we know it today. So this is the first report of Xylella in Central America. In our history, why did we study Xylella fastidiosa? Is because around the 1980s, the 1990s, coffee producers started to report something they called crespera in coffee plants. Crespera means leaf curling. So it's like it's a mild symptoms that form this crespera disease, and the report is mild symptoms, leaf curling, asymmetric leaves, problems with shortening with the internodes, and some chlorotic issues. This is what we know as crespera. This is a mild version of what is known in Brazil as coffee leaf scorch. We don't get to that point where we see scorch. And really, it's such a mild symptoms that there is no real estimates of the Xylella fastidiosa impact on coffee production. And as we have done more testing, we know that Xylella is prevalent and present all throughout the coffee regions of Costa Rica. And if I took you to a coffee plantation here, we can describe Xylella as colonized asymptomatic. This meaning that if we start to look for Xylella in these coffee plantations, you will find it in more than 50% of the plants, but plants usually don't have symptoms. Usually, if we see some crespera disease, it would be less than 2% of some of these coffee plantations. So there is really no estimates or really impact of Xylella in coffee plantations. There are other diseases like coffee rust that really have a drastic impact on coffee in these tropical conditions. One thing that is needed to remember is that coffee is not a native plant of the Americas. This is also an introduction, and it's an introduction near the end of the 1700s. This is a photo of around the 1900s. And this is important because if Xylella fastidiosa is endemic in these conditions, coffee resulted as a susceptible host to Xylella fastidiosa. But in our conditions, it is not as drastic. Probably, as I mentioned before, it has to do with a lot of other factors in these tropical regions related to climate and stable conditions that may favor the not presence of drastic versions of Xylella fastidiosa in Costa Rica. But it's really important to remember that coffee is also an introduction in Central America. After this, we started with coffee. That is what started most of the study of Xylella fastidiosa in Costa Rica. As I mentioned, most of the plants we find as positive are colonized plants, mostly asymptomatic. We have also found Xylella in other grapevines that have been planted throughout the years in Costa Rica, in coffee, in citrus plants, orange plants, usually used as shade in coffee plantations. This is also true for avocado. And we have also seen, and this one really is important because we have seen or detected Xylella fastidiosa in Nerium oleander that is used as ornamental in houses in Costa Rica. And there is a relation between Nerium and ST53 types, that is the Pauca one that is circulating in Costa Rica. So from our experience, we have seen that these Nerium plants can really get the disease, show disease, as well as Vinca plants. And this is one of the reasons we use these plants also as like sentinels. When we go out for looking for Xylella, we try to look for these plants as a starting point in some of these locations. This is just to show you an idea with using immunofluorescence. We have tried different plants that are important economically, and usually this is what we get. We get that Nerium, Vinca, or Cataranthus, and coffee plants usually are positive. We have found another Xylella in different plants, and we are starting to look for more native plants to work on. This is some sampling sites we did it for these years, and as I mentioned, we use these sentinel plants around the country, and we had 30% positivity of all the plants we screened. But as you can see, the red dots are negative plants and are more in the lowlands. With the Basil initiative, we are going out and trying to do a complete screening of the country to find out if really there is a relation between Xylella and highlands in Costa Rica, or is that we haven't looked enough in the lowlands to see if we can find and try to detail more where Xylella is focusing in the country. This is a historic accumulation map of all the isolates we have eventually worked on and successfully been able to have the bacteria to work on, to characterize, because this is part of a difficulty of working with Xylella, but also very important to generate knowledge of circulating strains and how bacteriology they work, and what is happening with the diversity of circulating strains in Costa Rica. We're looking for more strains, and we're trying to leave the central part of the country and try to have isolates very distant or isolated from the ones we have already succeeded on having on our collection. From that information, with the isolates and with collaborations, it is known that our strains, here are our Xylella fastidiosa strains, show more diversity when whole genome sequencing analysis is performed than Xylella fastidiosa strains from the south part of the United States or California. And it's also true that some strains that are Xylella fastidiosa from Pauca are related or closely related to the Italy ST53 strains. So this is using whole genome analysis. And so we can, if we have to tell about our circulating strains, we can say that we have a Xylella fastidiosa, subspecies fastidiosa, endemic strains that represent a unique population with broad genetic diversity, as shown here in this picture. And a Xylella fastidiosa subspecies pauca that is an introduction from South America. This conclusion that pauca is an introduction from South America comes from studies performed with other groups that show this relation from pauca from Brazil, that is what is predominant in South America, that maybe some of these strains moved into this Xylella fastidiosa environment that we see in Central America with great diversity, and that we know that this is possible an introduction because when we go out to the field and all the isolation attempts, what we get is Xylella fastidiosa and just one particular strain of Xylella fastidiosa pauca in our conditions. This is when you perform MLST for determining the sequence type of our Xylella strains. These sequence types, if you look at the database, is very unique. These STRs are unique to our isolates. And as I mentioned, we see a lot of great diversity of isolates. The one thing that we have seen with using MLST for this purpose, when you have a lot of circulating diversity, is, for example, we know by whole genome analysis that the strains are different, but since this MLST analysis is limited to 7 genes, we get these types of things that we know that are not true, that we can get to ST, for example, this with the asterisks, to ST77, but we know that phylogenetically they're distant. So these are things that may happen when you have a great diversity and using this technique. For this, we have also used microsatellite analysis, short sequence repeats, and with this we get a more a better picture in relation to whole genome analysis than what we get with MLST. And with this, as you can see, we here put we put the microsatellites as barcodes. Each bar is a microsatellite. And as you can see, with our Xylella fastidiosa strains, we have a lot of diversity within the strains. But with the Xylella fastidiosa pauca that we have characterized, we see that usually they're practically the same. This meaning that the circulating pauca strains have less genetic diversity than the fastidiosa strains, and this may be related to the endemic origin of Xylella fastidiosa and the introduction of the pauca strain. Another thing that we have seen is that, at least with the work done up to today, is that there is no correlation between geographic location and genetic traits of isolates, meaning that we don't see patterns of ST, at least for STs or whole genome analysis, we don't see patterns of the presence of some type of ST within a specific geographic locations. We find ST types throughout at least the coffee regions we have studied. We also find this is near the university. These are still urban coffee places, coffee plantations. And as you can see, this is 1 kilometer by 1 kilometer. If we go to the same coffee plantation, we can get a lot of genetic diversity within very reduced space of in distance. The red ones here are very important because these are the ST53 strains, isolates, and these are the ones that have been related to the Italian outbreak. And interestingly, these ones are usually related to nerium, to oleander trees. So there are questions that we have around this, is how similar are these strains phenotypically? What are the genetic difference among the strains? How close are we of uncovering the total diversity present in Central America in regards to Xylella fastidiosa? What is the pathogenic potential of these strains? Since we don't live with problems in our agricultural crops, but we know that the recombination that may be that is held in these conditions may give some potential pathogenic to these strains that may cause some kind of epidemic or outbreak in our conditions eventually, but as we have seen in other latitudes where other conditions are different and may favor the development of Xylella disease. And what about the vectors related to these conditions in our country? We have done some phenotype work. This is just an example. We have done Raman spectroscopy to characterize our strains. And what we see is there is no relation between genomic and phenotypic characteristics. This is true for some of the phenotyping we have done for these isolates. We have also done biofilm assays to see the potential formation of biofilms of some of these strains. And also here, there is a bit of correlation between some genomic strains and the potential of producing biofilm. But one interesting thing that we have seen is that the plants that have shown symptoms usually are the ones that are not so productive of biofilm in vitro conditions, at least. We have done mechanical inoculation assays in using coffee and vinca isolated strains in grapes and oleander, and that what we see is that these Xylella fastidiosa strains are not or do not show preference for these hosts. Regarding potential vectors, there has been some work done in Costa Rica, especially because in the biodiversity field, because in some point there was a movement or initiative to do a catalog of biodiversity in Costa Rica. So there was a lot of prospecting and classification of species. And this is a paper of potential subfamilies of psychedelics that could be eventually vectors of Xylella. And some works of genetics throughout the years have been also performed. However, vector-specific Xylella fastidiosa transmissions have not been performed until we started doing this some years ago. And we went out. This is some work that we have been doing throughout these last years. We have accounted the absolute and relative abundance of psychedelics in coffee plantations. We know how many individuals per plants to expect and how many species per plant as absolute numbers. So you can see 4.3 and 3 is the numbers. And we have also relative data regarding the main insects or psychedelics that we have that are related to coffee. We take this to the lab, and we have also done through artificial transmission assays detection of Xylella from the feeding after they feed. 24 hours later, and we see that 50% of these vectors, of these insects, cicadelids that we collect are Xylella positive. And we have done the transmission assays on plants, on coffee plants, and we know that from these 6 species, 4 have we have detected positive plants 6 to 1 year after transmission assays were mounted. So we know now that this information that there were potential vectors, and we know at least from our work that we have 4 vectors that are really true vectors of Xylella fastidiosa. One of the take-home messages is that our conditions, our environmental conditions are different, and this is has to do with the way this potential pathogen is behaving, and why it is not impacting our, at least for now, our coffee crops or plantations, especially coffee, but other potential plantations that can be affected by Xylella. And is that, although, as I mentioned before, as we see the coffee plantations, but as you can see behind this whole land of coffee plantations, the mountainsides you see here are primary forests. So one of what we think is that really, although we have an agricultural environment, when we go to the field, we find still a lot of diversity within the strains. We find a lot of diversity of vectors, although it is a monoculture. So really what we are seeing is that the biodiversity that we have surrounding our economical plantations still have a big pressure on this system that maintains it like a very ecological ecosystem where diversity is still seen and pushes, possibly drives it to not fix a potential efficient vector or fix some more pathogenic strain that would give us eventually consequences of disease. So it's really susceptible plants, but with a very healthy environment that is keeping these conditions. And we don't see any or very few symptoms and problems in our production, at least in coffee. And this is the representation of what happened in Italy with a strain that epidemic, very aggressive. And what we see here in Costa Rica, non-epidemic with a lot of host range and this potential and these strains are really not so pathogenic and diverse. So this is what I show you here. We have a lot of these strains, but I want you to put attention on these red dots. These are the ST53 that are related to oleander trees in Costa Rica. And these are the ones that are similar to the ones from the ST53 strain from Italy. So this would be 2 scenarios where you have ST53 in oleanders that cause disease, but because we really see that these trees get disease, they're probably more pathogenic than the strains from coffee. But these strains in another conditions with other abiotic factors and with some changes in the pathosystem, the vector and the susceptible host gave us very drastic results. And this is the relation between our strains in Central America, especially the STT53 and the Codiro Tedono strains from Italy. that, well, as you know, that the common knowledge is that all this happened through plant trade. And this is a relation with the outbreak in Europe, the Italian outbreak, as more knowledge on Xylella in Europe has been established. It is well known that plant trade, not only from Central American regions, but from North America and South America, has also contributed to the presence of other outbreaks in other countries in Europe. When this happened, the Xylella fastidiosa outbreak in Europe, just to let you know that this had other impact in Costa Rica, not because Xylella affected our agricultural crops like coffee, but there was a very lucrative business that was exporting plants, or inverse, importing plants from Europe, from tropical landscapes. And this one, when the outbreak came out, there was a stop on this type of business, and this had an impact on Costa Rica, because a lot of people lost jobs that were related to this lucrative business. But everything evolves, and I tell you that in recent years there have been efforts between the plant pathology authorities, at least in Costa Rica, and I know in Mexico also, and private companies that pursue free status of Xylella.
Ah, vais pôr o tradutor automático.
And
Não, alguma
just,
palavra, isso.
I'm just finishing the importance of surveillance in endemic surroundings.
Ir à reunião,
We
cancelar.
have high prevalence of Xylella in plants, broad diversity of circulating strains, broad diversity of vectors, environmental and agricultural practice that may favor conditions for the development of outbreaks. So if we do surveillance on how healthy are our conditions, we can eventually have predictive values of if we see that something is changing in the environment. And this is what I had for you to show. We have a lot of questions. There's a lot of missing pieces, and this may mislead to interpretations. And if we complete the gaps, we can take better decisions, not only in our countries, but in other countries that depend on and rely, or we need for economic purposes in the future. And that is the presentation I had for you for this afternoon. Thank you very much.
Thank you, Carlos, for your complete frame of the situation in Central America. I have a couple of questions about your presentation and about the perspective for the future of Xylella fastidiosa in Central America and in the world. First, I'd like to know your opinion, if you, considering your knowledge about the biology, distribution and spread of Xylella, what you consider that European countries should take to account to reduce the impact of the bacteria?
Yes, I think we need to keep on knowing, on exploring and doing research on diversity. We are far from knowing how much of the genetic pool of Xylella fastidiosa, this fastidiosa diversity we have detected. And this is very challenging because isolation of Xylella in tropical conditions is more difficult. We have a lot of endophytic bacteria that make very difficult to isolate the strains. So that is one thing. And two, we need to keep surveilling our conditions, because in the moment we find a shift, and we start to lose diversity in some crops. That is an alert, because that would mean that there is a better vector or a better strain that can fix, and that it could have a more potential, pathogenic potential than other circulating strains that we know. So there's a lot to be done, and I think our main contribution is in relation to the biologic pathosystem in our conditions as a predictive value for eventual outbreaks here or in other latitudes, as we have seen throughout the history.
Okay, thank you. And also, do you consider that European countries should understand the Latin American management problem, or we are in the same situation?
I think the situation is a bit different. We need to, as countries that want to do business with Europe or other North America, it is important that our plant pathology authorities have improved our system to be more rigorous, so we can have all the business or economic issues regarding plant movements and plant business without any trouble. This was a big problem when the Xylella outbreak started, but I know that there are efforts going on where the plant pathogen authorities and companies interested in plants business that have to be moved are doing very big efforts to go in line with all the European conditions, so they don't have any problems and could keep going on with their business and their field of productivity. So
Thank you so much, Carlos. There's no more questions, so thank you so much for your presentation. Now you're going to pass to the next two speakers, Paulo Garcia, the Subdirector General of Directorate General of Food and Veterinary, and the engineer Clara Serra from the same institution. The DGAV mission is the definition, execution, and evaluation of the food safety, animal protection, animal health, plant protection, and phytosanitary policies. We invest in the functions of the national in Portugal. The DGAV, as the National Phytosanitary Authority, defines and coordinates the National Survey Program for Xylella fastidiosa. So, Paula Garcia and Clara Serra are going to tell us about the Xylella fastidiosa in Portugal, the national plan to eradicate the bacteria and control the vectors. Paula and Clara, you can start.
Hello, good afternoon. Thank you very much. We are here in Lisbon together in the same room. I have prepared my presentation, specifically my presentation in Portuguese, so because I expect to be able to speak in Portuguese, but I know that I will now present in English. Sorry for that, because the PowerPoint presentation is mainly in Portuguese, but I will try my best to try to translate the PowerPoint presentation into English. Okay, my colleague Clara, she will be presented in English because she was more attentive to the program. So sorry for that. So we will try to share our PowerPoint. We have two PowerPoint presentation, one related to the general overview of our national action plan, and the other presentation that will be done by Clara will be specific for vectors control and monitoring of potential vectors, Xylella fastidiosa vectors here in the country. So let me try to share the presentation. So I think you are seeing the presentation. Yes, can you confirm?
Yes.
Okay. So we are talking about, we are going to make a very short presentation regarding the Xylella fastidiosa in Portugal and the national plan to eradicate the bacteria and also control the vectors. So as I have mentioned, I will present the first part of our presentation related to the national plan. And then Clara will talk about the control of the vectors and the monitoring program regarding the vectors. So as everybody knows, we are a member state of EU, so we follow the European Reglement regarding the pest control. So we have a new reglement established at EU level that we need to follow. So they are harmonized measures applied by all the phytosanitary authorities of the member states, how they control, how they make the surveillance, and how they apply the measures to eradicate the outbreaks of quarantine pests. So this is the Reglement 2016-2031, published in the 2016 and enter in vigor in 19, I think, October 19. So this is a new framework, legislative framework that it's completely different from the previous one, and it's more harmonized. So we have in this legislation so-called priority pests. It's 20 pests that we have defined at EU level as the priority ones. What is these priority pests? Mainly they are insects, as you can see here in the picture. We have here the picture of each one of the 20 priority pests, and you can see 17 is insects and the nematode, pinewood nematode, and of course Xylella fastidiosa is also included in this list of priority pests according to the EU legislation. And the priority pests are the ones that have been established by a specific regulation of the Commission that established the list of these 20 priority pests. And according to the study that have been done by the Joint Research Center, it is an institute of research belonging to the European Commission. They have considered several aspects: social, environmental, economic aspects of impact of the quarantine pests in the EU. And according to their study, they have established these 20 priority pests that we need to have a different approach, a more strict approach regarding their control, their surveillance, and also we are obliged to have contingency plans in order to be ready if there is an outbreak of one of these quarantine pests, of these priority pests. So priority pests are, of course, quarantine pests according to the definition of the European legislation, but inside of this big list of quarantine pests, these 21 are the ones that their potential economic, environmental, or social impact is the most severe in respect of the Union territory. And as I have mentioned in the previous slide, we have mainly insects and of course also the nematode, the pinewood nematode, but Xylella fastidiosa is also included in this one. In this slide, we have here the lists of these 20 priority pests, and in bold we can see 3 of them are present in Portugal. So one is Bursophalangus xylophilus, the pinewood nematode we have in Portugal for several years. Popilia japonica is an insect present in Azores Island, not present in the Portuguese mainland. And unfortunately, we have now also Xylella fastidiosa in the country, several outbreaks, and we will give you some update about the situation in Portugal regarding this priority pest, Xylella fastidiosa. So, as you know, Xylella fastidiosa have a huge amount of host plants, and there is a distribution around the world, not mainly in the American continent, but also several outbreaks in Europe. In Europe, France, Spain, Italy, and Portugal are the only countries in EU that have outbreaks of Xylella fastidiosa. And all of these countries, Portugal, Spain, Italy, and France, are dealing with eradication measures in order to avoid the spread of these bacteria. And also in certain cases, like in the islands of Balearic Islands and Corsica, is already in containing, as in Apulia region, because eradication is no longer possible. But in Portugal and in France and in mainland of Spain, we are still applying the eradication measures in order to try to eradicate these bacteria in the country. But perhaps in certain cases will be no longer possible to do that. So the phytosanitary measures that we used in order to elaborate the contingency plan, because as I have said, for the priority pests, we are obliged to have a contingency plan. So we follow several documents. And one of the documents I have here in this slide, some of the documents elaborated by EFSA and published by EFSA that contribute to establish the harmonized measures at EU level for the eradication and containing of Xylella fastidiosa. So we have here the scientific opinion on the risk of plant health posed by Xylella fastidiosa. It was a scientific opinion published by EFSA, and also EFSA has updated the scientific opinion on the risks to plant health posed by Xylella fastidiosa in EU territory. EFSA have a huge role regarding the establishment, the help to the member states and the Commission to define the measures, the legal measures to be applied by the competent authorities. Normally, these measures are supported by scientific opinions, namely by EFSA. Also, we have regarding the documents produced by EFSA, we are now following the pest survey card, and also we have guidelines for statistically sound and risk-based surveys of Xylella fastidiosa. So the work that we are doing here in Portugal are follow the same approach, the same statistically guidelines by every competent authority in EU. So we follow the same approach for surveillance and also for the eradication measures. So this harmonization is very important, and we have it in the case of Xylella fastidiosa, establish a specific reglement of the Commission that established those measures that need to be applied in a harmonized way by all member states, not only for surveillance, but also for the eradication and also for the import control, for instance, at the border ports, in the airports and border control posts, in control of imports plants for certain countries. Plants for planting are also controlled in the several border posts by the phytosanitary services of the competent authorities in the same way, in the harmonized way. So this is very important in order to ensure that all the member states are applying the same rules in order to protect the European territory. So this is a map that is included in the opinion of EFSA, and indeed, Xylella fastidiosa is because can cause a very negative impact in the EU production value, mainly in olive trees, but not only almonds and other several agricultural crops very important for EU. Is estimated that the loss produced by Xylella fastidiosa, if it is spread around EU, can cause a annual production loss of 5.5 million euros and can affect 300,000 jobs across Europe that are currently involved in the production of the main crops that can be affected by these bacteria. So here we have 3 maps. Each one represents the risk map according to the climate suitability regarding the subspecies of the Xylella. The first one is the estimated climate suitability for Xylella fastidiosa, and the one in the center is for Xylella fastidiosa subspecies multiplex, and the other in the right side is the map regarding the Xylella fastidiosa subspecies pauca. And you can see the different colors, and then we have light yellow, for instance, or light blue is the areas in Europe in more risk that have more climate suitability to be affected by Xylella. And you can see Portugal that mainly for fastidiosa and for fastidiosa multiplex have a high risk for this disease if there is no control of the disease in the country, because we have the good conditions to enter and spread and established of these bacteria in the country. So this map is very important regarding the risk, and that's why we are facing with several problems, and that's why the outbreaks are mainly in the southern countries of EU, France, Italy, and Spain, and also Portugal. So what we do for surveillance and eradications, as I have mentioned, we have harmonized procedures in order to perform the eradication, surveillance, but we have a contingency plan. This one in the picture is the last version, 2022, but we have a contingency plan for several years. I think the first one was in 18 or 19, but we try to update this contingency plan according to the scientific information that we have, the most recent. So we have updated our contingency plan for Xylella fastidiosa and their vectors. And the contingency plan includes information regarding the legislation, about the bacterium, about the vectors, what are the measures, the preventive measures to avoid the introduction of the bacteria and spread, also includes the actions, the measures for surveillance, how we do the surveillance here in Portugal, and also the organizational structure, because we need to have to be ready to act when we have an outbreak, and also the definition and identification of the laboratories that are designated by our services to perform the analysis of Xylella fastidiosa. Also, the contingency plan also includes the actions that we need to follow if we have suspicious of the presence of a bacteria, and also the measures that we need to apply in case we have the confirmation, laboratory confirmation, of the bacteria. So this contingency plan is very important because includes all the information related to surveillance and also to the measures that we need to quickly put in place when we have outbreaks. So our national survey program started in 2014, just after when we received the information about the outbreak in Italy. That was in late, sorry, I need to drink some water. So we have received the information of outbreak in Italy in late 2013. And we started to perform a surveillance here in the country, in Portugal, to know if, sorry, again, I need water. So this surveillance program is coordinated by the competent authority, is our service, General Director for Food and Veterinary, and is up to the regional services of agriculture and also up to the Institute of Forestries to implement the inspections and sampling in the country. We have identified places where the risk is higher, and these places are normally the nurseries, the garden centers, and also the orchards of the main crops that can be affected by Xylella, such as olive, prunus, citrus, vitis, and also, because it's very important to Portugal, Quercus suber is one of the most important species, forest species we have. It's cork tree that it's very important to Portugal. So we have identified the places, the high risk places where we prioritize our surveillance, but we also include urban areas, public gardens and private gardens, and also the highways and other places, not only agricultural forestry places, but as we call it, natural environment, are also included in our surveillance. For this, we follow the pest survey card that it was a very recent publication made by EFSA, and we start to apply this year because we have a derogation. But since 2023, we are now obliged to use pest survey card also for surveillance at national level. So this pest survey card is very helpful because gives a more harmonized measures how to perform the surveillance with a very statistical sound procedure in order to know if we are doing the good surveillance or not. So this is a very good tool that we have now coming from EFSA, and EFSA is now established these pest survey cards, not only for Xylella, but also for other quarantine pests that we also are going to use this pest survey card, sorry. So we have the contingency plan. We do the surveillance in the area at national level, and in case of detection and confirmed by laboratory, we need to act quickly. And in this case, we elaborate the eradication plan for each of the demarcated area. So each demarcated area has a specific eradication plan because depends of the area, depends of the kind of crops, if it is an urban area, if there is a forest site. So we need to elaborate an action plan to eradicate for each demarcated area. So the first action plan that we have established and elaborate was the one that when we have the first outbreak in the urban area of Porto in the north, in the north part of the country, where we have in 2019, in January 19, the first outbreak of Xylella in a lavandula species. And we have established this action plan for eradication. And of course, each year, the action plan also is updated according to the situation of each demarcated area. So each demarcated area have a specific action plan that specify the area that we need to, all the measures that we need to apply and to the actions that need to be taken in each demarcated area. So the action plan defines the infected areas. So infected areas, we need to demarcate the infected area and also the buffer zone. Our action plan for eradication also includes all the information regarding the inspection and also sampling. So normally there is a more intensive inspection and sampling in the demarcated area. We need also to consider it in the action plan, the ways and how we can uproot and destroy the plants inside the infected zones. And we define also all the restrictions regarding the productions and the movement of plants for planting that are applied in both in the infected zone and also in the buffer zone of the demarcated area. And also it is forbidden to plant the plants of the specified plants in the infected area. Is no longer possible to plant the same species that have been found infected in the infected zones. So this is several images of our platform, IT platform that we used to make the management of the demarcated areas. So when we have an outbreak, an infected area of 50 meters are established, 50 meters surrounding the infected plants. And in this area of 50 meters, we need to know we need to have the list of all the species that are there in that area. We need to have to identify all the species present, destroy the species that have been found positive, and we need to sampling all the species that are hosts of the bacteria, not only the ones that we need to destroy are the ones that are infected for the subspecies that have been identified, but the other species present in the infected area need to be also sampled. Then we have a first buffer zone of 400 meters where we apply a specific it's difficult to say this word, sorry. Ribes Plus, it is the program developed by EFSA, where we perform our sampling in a more intensive way. And then we have the buffer zone 2. We call it buffer zone 2, is the area between 400 meters and 2.5 kilometers from the infected area, where we have also a need, where we also apply Ribes Plus, that is the program for the statistical sampling program that we are obliged to apply in order to be sure that we are doing the right sampling in order to establish the demarcated area. So this is performed each year. And we need also to inform not only the farmers, not only the nurseries, not only the operators that are in the demarcated area, but also the population in general, because there are, as I have mentioned, some restrictions regarding the movement of plants for planting from outside the demarcated area. So we published the so-called despachos, that it's a formal notification from our services that define the measures to be applied in each demarcated area. So this document is in our website, and also is it's very important to be communicated to all parts in order to be sure that the people inside the demarcated area are aware that they are in the demarcated area of Xylella fastidiosa, and some restrictions regarding the movements of plants for planting need to be applied. So these despachos are updated because when we find a new positive plant, a new infected area, a new infected zone, this is the red bullets, the small red bullets are the infected zone. This is the demarcated area of Oporto. And we can see here the red bullets are the infected zones. And then the black line is the buffer zone surrounding all these infected zones, these red dots here spread in this demarcated area. So we update this information because when we find a new species in the demarcated area, or we find, for instance, a new subspecies of the bacteria, or there is an enlargement of the demarcated area, we need to update this information. So in the demarcated area of Oporto, we have already identified 78 plant species that have been found infected by the bacteria. So there is, we also published this list. This list is updated. This one was updated in March, in the beginning of March, because we have found a new, this new one species, this in bold. It was the last one found positive. So unfortunately, this list is being increasing. And you can see here a lot of spontaneous species that we have around the country are very common in the natural environments. But also you can see here very important agricultural species like, for instance, vitis, and also Olea europaea. And also, for instance, we have here Castanha sativa. It's not, I think it was the first time we had Castanha sativa infected in the last one was in Corsica, I think, the only one. And also we have a very bad news last year with several citrus species also infected by the bacteria in this area, in this demarcated area of Oporto. So this is a very concern. This is a very serious situation because it's very difficult to deal with all of these species because a lot of them are spontaneous, are everywhere in the area. So it's very difficult to deal with this, to apply the eradication measures when we have such a huge list of infected species to be taken into account to apply the eradication measures. So now, this is a very difficult slide for me to explain because we have now a very complicated situation in Portugal. We have now at the present 18 demarcated areas, seven in the region of in the north of the country, 8 in the center of the country, and 3 demarcated areas near Lisbon Valley. So this is a very complicated situation. and the subspecies of the bacteria already detected in all of these demarcated area. Of course, some of them, the majority have just one subspecies, but we have already found multiplex ST7. Multiplex ST7 is the main subspecies found in the Oporto region, but we have now more recently fastidiosa ST2 and fastidiosa ST1. So this is a very worrying situation that we are facing. So the major agriculture and forestry species that we found positive are olive trees, almond trees, citrus, vine, castanha, oak trees, and peach trees, and rubus. So this is the main agriculture and forestry species that we are finding positive in this demarcated area. I just want to clarify that we have these 18 demarcated area now, but the majority of them, we have only one positive sample there. And we are going now to make all the actions that I have explained in the case of the Porto region. We are going now to perform the intensive sampling in those areas in order to know what is the situation in that new demarcated area that we found one or two positive samples. The majority of this demarcated area, we have found positives in nurseries when we, because there is an obligation to test some species before the issuance of plant passport, before moving the plants for the internal market, and some of these outbreaks we have obtained when we receive the results from the lab in the scope of this control of the plant passport. Also, I would like to mention that we have 2 or 3 cases that we decide not to go ahead with the demarcated area. It's possible not to demarcated areas. The European legislation have a derogation to the member states that can derogate for the demarcated areas when the plants arrived to the place already infected. So we have plants, and 2 or 3 cases where we found plants coming from other member states just arrived to the garden center, just arrived to the nursery, and when we collect the sample, the plants coming from other member states were positive. So in this case, we did not establish the demarcated areas in that cases. So this is the situation in Portugal. You can see here the Portuguese map on the left side, and you can see the bullets, the small dots in different colors, where we have now the demarcated areas that we are working in order to apply the eradication measures. But as I have mentioned, the majority of these demarcated areas represents only one single plant positive. So, and now we are going to check if there is more or not in the demarcated area that have been established, and try to understand why this is happened now. We don't know if we have a more surveillance, a more strict and complete surveillance program than the other member states. We don't know, but we are facing with a very complicated situation. But as we have in this demarcated area, the majority of them correspond only to one positive sample. We hope that we can eradicate this demarcated area easily. Let's see what will happen this year. So what are our major concerns? What are our major desafios?
Agora igual.
So we think that we need to, in order to establish a better phytosanitary control, we need to have more and better training and information. We need to have information about the subspecies that we have in Portugal, where they were come from. We have also, we think that there is a need to improve the intra-community movements of the plants for planting, the plant passport regime. Also, it's necessary to work with the nursery sector in order to have more professionalization of this sector. It's important to have a reinforcement of agricultural advisor services. It's important to have this information to the farmers and also to the nurseries in order to help them to prevent the spread of quarantine pests, specifically Xylella fastidiosa. Also, it's crucial to have a rapid intervention capacity for the destruction of the infected plants, for the sampling from the demarcated areas actions that we need to perform, increase the diagnostic capacity. As you can easily understand, with such a number of outbreaks, we need to have a huge capacity of the laboratory to test the several hundreds of samples that we need to take. Applied research is fundamental, and last not the least, or least not the last, vectors control. So vectors control is the key point that we need to improve, and we need to have more information, more technical information in order to control the vectors. If we control the vectors, it's easy to control the disease. So, and now I will pass the floor to Clara, that will talk about the vectors and the work that we are doing here in Portugal regarding the monitoring and vectors control. Thank you. And sorry for the presentation, that it's a mixed in Portuguese and English. I don't know if you want to have some questions, to put some questions or Clara can present also the vectors, and then we can perhaps the questions at the end.
We can ask in the end.
In the end, okay.
Of Clara.
So Clara
okay.
will now speak about the vectors, okay?
Can you see it and hear properly?
Yes.
Yes. Thank you very much. Okay, I present myself, Clara Serra. I belong to the DGAV for Plant Inspection Services, and I'm responsible for the Xylella program to see how to implement the surveillance and eradication of program for Xylella. So considering what my director said, Paula Garcia, there is a lot of new infected zones in Portugal, and I will speak about concerning the vectors. I'm not a specialist, I'm not an entomologist, but I will have some revised information, and I want to share with you. We know that the potential vectors in Europe, they exist all over Europe. They are insect that is spread all over the European continent. As you can see in this map, there is certain species, and you can see Philaenos spumarius and Cicadella viridis and Afroflora alni and so on. So these vectors are potential vectors, although Philaenos spumarius have been reported the main vector in demarcated areas in Europe. So until now, we have this main vector. Of course, in other demarcated areas, perhaps another Neophilaenus can play a role as a vector, but considering the importance of the vector, Philaenos vector, like the colleague of yesterday say, may be the vector. That is what. So we have these potential insect vectors. This is Neophylaenus, Philaenus fumarius, Cicadella viride, Cercopsis, and Afrophora. So all these vectors are potential. So they exist in Europe. They have maybe different distribution within Europe. And of course, for example, here in Portugal, we have a lot of Cicadella viridis related to vitis plants and so on. So what Philaenus fumarius is a very known insect here for even for the common citizen, because they can see the splits in the plants, which looks like splits from humans, but it's not, it's from the insect to protect themselves during that nymphal stage. And according to what you can see, the biological cycle, these vectors, these adults will go to make normally place the eggs during November and February, and during this time they will place the eggs during November, December, and they overwinter as an egg phase. And when you see other Xylella problems in other countries, like in South America, maybe the vectors are not overwintering in a egg phase, they are adult. So this is something that can help us in cutting down the cycle of Xylella, because all the new generation of vectors that will appear in February, March, they will born clean, at least clean as clean adults, and they will be infected later during their probing activity and feeding activity. So this is a very important knowledge, because if we want to make a cut in the population of vectors, we have to try to catch them in the egg phase, which they are not flying, and that maybe gives you the chance to interfere in their life cycle. So that is one important aspect. Relating to this, after they are going in the adult phase, they can move around in different environments. They can go to shrubs, of ornamental shrubs, or vegetative in natural areas. They can visit the crops of trees like quercus trees, olive trees, or other bigger trees. But so they can move around up and down in this type of environment. And this type of environment, you have many places in Portugal, they have this type of occupation. When you see urban areas connected to rural areas, you can have this type of environments. So therefore, one of the measures that we have in our action plan is to control what we call the places where the vector go to do the laying of the eggs. That is what we call agricultural practices that you should use, not only insecticide, but you can have mechanical ways to disrupt the cycle of the vector. So that means the vector can not get to the adult stage, and that is one challenge to try to get. So we have this legislation in force, that is Commission Regulation 2020-1201 from August. And we have to also this legislation at the Portuguese national legislation. So this is to make more enforced the measures in place. So we could use directly the legislation of the European legislation, but even then we place in the national legislation. So according to what Paula Garcia showed you before, we have in the north, we have in the total mainland Portugal, 18 demarcated areas. You know, Portugal have also highlands, Azores and Madeira, and they are 3 areas as well. At the mainland Portugal, we can find 7 demarcated areas in the north, 8 in the center, and 3 in the Lisbon Alto Tejo. I make here underline here in my slide in the bottom. The Algarve demarcated area was lifted last year according to specific derogation. We can derogate not to demarcate, or we can have a specific derogation. You can demarcate, but later you can consider that the demarcation can be reduced, and after reduction of the demarcation, you even can lift it. But that is a lot of investment you have to do in sampling. So we did during 2020 and 21, and we were able to lift the demarcated area of Algarve. So no longer is now a demarcation in Algarve. Although we have to have a 2-year surveillance program, that means we have more enforced program in the, let's say, free area of Algarve, but we have to have that surveillance specific, which is more intensive to guarantee that the lift was done in a secure way. So when you can see what are the host plants involved in this demarcation, you have Prunus, you have Piracantha, you have Salvia rosmarinus, Olea europaea, Hibiscus, Quercus spirinacis. Some of those plants are important crops, but you can see some of them, they belong to the environment, like Quercus ilex and like Fraxinus. They are plants, they are living in the wild in some rural and semi-natural areas. So we suspect of some spread to these areas, not by human-assisted spread, but by vectors. I think that is quite obviously when you see those type of outbreaks. So more detailed information on vectors we gathered in the North region from 2019 until today. We have some more detailed information concerning vectors. So this demarcated area of Porto, we have 168 infected zones. And in those zones, we found multiplex ST7 and fastidiosa ST2. The fastidiosa ST1 was found in the other areas that we demarcate recently in the central region. So that means we have a distinct distribution of ST1 and ST2 present in our country. So, and considering two vectors, so far, we have been detected infected vectors of Phylloenus spumarius in this area. And very recently, we found one sample with Cicadella viridis also infected. We don't know the role or the importance of Cicadella viridis in spreading the Xylella in this north region. We are more thinking about Phylloenus spumarius, that is the vector that really plays an important role. Cicadella viridis can be detect infected, but how effective it is to spread Xylella to other crops or to the vineyards, it's not, at least we don't have that evidence that it is a very important vector. So fine, now we have a lots of plants found infected, about 78 species found infected in the demarcated area. And you see most of these species are host of the bacteria and also host of the vector. So you have a perfect combination. In these plants, you have the Xylella, you have the host, and you have the vector. So that means the vector have many opportunities to get infected because he likes to go in these plants. So we have a very situation, very, let's say, very important to understand the epidemiology of Xylella in the region of Porto. And just to give you an idea, the Ulex, which is a kind of what we call a spontaneous plant, which is on the covering crops under the Quercus subor stands or forest stands, you have about 53 infected zones where you find these species infected. Salvia rosmarinus, which is an ornamental which is spread all over Portugal, and many people like to have these plants. Everybody can cut a piece of this plant and make a new plant. So now this is a very risk plant. If you take plants from Salvia rosmarinus that are in the Porto region, you are taking maybe plants which are infected because they are not removed properly. So these Salvia rosmarinus, and it is present infected in 70 of those infected zones. Lavandula dentata also another ornamental, which is very important and found infected in this area. And in 11 infected zones, we have Quercus suber, where it is, let's say, we have conclusion that is a natural spread by vector, because these Quercus suber are spontaneously regenerated in forest stands. They are not planted by man. So it's not coming from nurseries. It is coming from the activity of the vector infected in the area. And also the same conclusion we take from Acacia longifolia. And now we have a new species, which is this Argyranthemum future. This is ornamental. And we also found some vitis plants also infected. It's not vineyards. We don't have vineyards. We don't have big crops like you have in Italy, or the olive groves, or we don't have the Prunus dulcis groves like you have in Valencia, in Alicante. We don't have big areas of crop affected. We have urban areas, semi-natural areas with lots of species of ornamentals and of natural vegetation, what we call the maquis, which is the French called the maquis. We have many of those plants infected in that type of environments. So what we do, how to capture the vectors in the demarcated area, according to our plan, we use a sweeping net capture. And we use this sweeping net mostly in the ground vegetation, because normally in the crop and on the top of the trees, we don't find many population of the vector because it is warm. Normally the vector like to go to shadow areas or to more humid areas. So our sweeping capture mainly, it is at ground level. So we can catch a lot of these insects in those. And also a very important conclusion from our work of this monitoring of vector, it is most vectors that we sweep on the second, because we have two periods to do the sampling. The first period is when they start coming out from the nymphal stage, but we have a very second period for us is very important to understand the epidemiology, which is when females go to lay eggs by the end of October to December. And this ground vegetation is full of females ready to lay their eggs. And these females that are adults are already at the end of their life, and they were during all their lifetime eating and sucking in plants that they were positive. That means, so if we want to find positive vectors, this is the best time to catch them and to test them for Xylella. And those, what are these xylem feeders? Normally it is Philaenus pomadius, Cicadella viridis, Lepironia in this northern area. But we also kept some called Eushellis, which are a phloem feeder, which is not considered as a potential vector, although we want even to make sure if this phloem feeder cannot play a role in the dispersal of Xylella in this region. So now what I can show you here is that what are results of our monitoring since 2019. So, for example, in the 2 years, 2019 to 2020, in 131 infected zones, all of them had vectors. Somehow they were there, Cicadella viridis, Philaenus pomadius, and so on. And from those infected zones, we detect infected Philaenus pomadius in 11 of those infected zones. And those infected zones are located mainly in the counties of Vila Nova de Gaia and Santa Maria da Feira. And this is a very promising landscape for the Xylella, for the vector, and for the hosts. And then we have the perfect triangle for the spread. This is very, very concerned for us. That's why we have to be very attentive to these results. Of course, our monitoring of vectors, it is what it is possible. I know there is some groups, they will dedicate more specific to the study of the vectors, but this is the outcome of the National Action Plan. So in 2021, in 84 of the infected zones with vectors, they were present in all counties, but in 22 out of that 84, 22 of them had Philaeus spumarius infected with multiplex ST7. And in 2022, we have also done the monitoring, and in 37 of infected zones, we found 2 of them with Philaeus spumarius. So far, we only found in the vector the multiplex ST7. So because we have the Xylella fastidiosa ST2 in this area, we don't have yet found any Philaenus with the Xylella fastidiosa. But we found Ulex, which is the spontaneous plant, which have already Xylella fastidiosa, and that only can come there by a vector. That's almost sure. So, and then again, I wanted to say you that we have this Cicadella viridis, very isolated positive case. So I don't know what is the real meaning, the role of Cicadella viridis in the spread of this Xylella in the environment. And again, we have also established a specific program for a 50 kilometers radius in the vicinity of the demarcated area. And there we didn't find any, we found vectors, but we didn't find infected vectors in that 5 kilometers surrounding of the demarcated area. As you can see here in the map below, you can see a green, a light green band, which is the 5 kilometers vicinity of the demarcated area. We make some sweeping around and we didn't find, we found vectors, but not infected. That is some relief. We maybe if we find a positive there, it should be many more difficult to justify. Okay. So what is the now what we have? Just an example, we have a platform called WebGIS platform for Xylella, and this platform allows us to have all the sample that is done in the Porto region and in the other demarcated areas as well. But this, it's only one example of the screenshot of that platform, where we have all the demarcation, all the infected zones have a code. And here I select 2 particular infected zones where you have Philaeus spumarius infected. And in that infected zones, what are the plants that are infected? Laurus nobilis, Ulex, Frangula, Hypericum, Erica, Acacia, Lavandulas. And so you can see what I've said before. We have a lot of spontaneous plants together with other ornamentals infected, and also with the vector. So that's why eradication measures have to take place in these infected zones. Otherwise, the vector will have always a host that is positive to give again a new generation with more Xylella in the vector. So, conclusion for our study in the demarcated area of Porto, we can conclude that there is natural spread by vectors into plants located in several areas, especially in forest and semi-natural areas. But in 2019, when our first year of the demarcation, we identified some infected zones that were related to plants coming from a municipality. Normally they produce plants for the municipality to put around the municipality, and those plants were infected. So that was in that time a way to disperse very quickly the plants infected. But of course, those nursery is not anymore on production, and there is a lot of restrictions for movement and for production for nurseries or garden centers in the demarcated areas. There's a lot of restrictions. So now coming back to the mainland, what we do in the rest of Portugal, what we do there. So we have some regions at the north center and Lisboa, Val do Tejo and Algarve. We make also some sweeping along the free mainland. We find Philaenus spumarius all over. We found Philaenus tessellatus mainly in Algarve region. We found Philaenus campestris mainly in Algarve. Cercopsis and cicadellas, they are all over around, and the Ochelis also have some regional localization. Normally, in the mainland free area of the national program, we don't have many information on the Alentejo region, but in Azores, we have Philaenus spumarius and Madeira cicadella viridis. But in Alentejo region, I used some information that we can provide you, that you can look at deeply. There is a work, a dissertation of a colleague from the University of Lisbon. You can see here the document, and that you can see in Alentejo, it's very important, is the Philaenus tessellatus, more than spumarius. And that gives you one idea of risk assessment of the Xylella in this region. Probably if Philaenus tessellatus is not a very good vector, probably we have less problems. But anyway, this is important. The studies on vectors, as Paula Garcia said, it's very important to know the vector behavior and abundance. Also, there is another work on Alentejo region that's, well, again, it refers that tessellatus is a very important species in the south, and you can see it go until Morocco, which is a different vector. So what is the possible role of Philaenus tessellatus in Xylella, which, okay, we don't have yet any information about being contaminated, but it is to be aware, it's a xylem feeder vector. So I think I finished my presentation. Thank you very much. And we can share some questions.
Thank you, Paula and Clara. Thank you for the overview of the situation in Portugal. I have questions, and I think that the participants in YouTube chat will also have questions. So my question is, as you know, there's a great concern for part of the production sector about the situation and possible impact of Xylella in your country. Please give us one idea about the next steps for prevision about the evolution of the bacteria in Portugal.
It's a very difficult very difficult answer. So we really don't really understand what is happening here in Portugal, because we are doing the surveillance as we have explained, using all the guidelines of EFSA, using the nurseries in Portugal are obliged to test their plants. There are six species that are obliged to be tested before the issuance of plant passport, and we oblige those nurseries to do that. And that's why we are finding these positive cases. We really, I don't want to say that because it's not a very political it's a political sensitive declaration, but I don't know if the other member states are doing the same work as we are doing here in Portugal. We really don't know. I think even if we have the risk, as you have, I have shown you the risk maps, Portugal is one of the countries that can be more affected by Xylella because we are in the risk, higher risk of enter and spread of Xylella. I don't know if there is such an intensive surveillance and also obligation for testing in the nurseries as we are doing here in Portugal. We 2 or 3 cases, as I have mentioned in my presentation, we found that the plants coming from other member states arrived to Portugal already positive. So this means, does it mean that perhaps the issuance of plant passport, it's not done as it should be? So we don't know. Perhaps also there is of course, in the future, what will happen in the future, we hope that the majority of these demarcated areas, as we have explained, represents only 1 or 2 positive plants there. We are trying to do our best in order to do what we have done in Algarve outbreak, in order to eradicate it if there is no other positive plants surrounding 2.5 kilometers from the infected zones. So let's see what happened in this survey campaign, because we have already started the sampling this year and also collecting the insects in order to know if there is insects or these insects are infected or not. So we cannot say what will be the outcome of this work. Let's see. But we sincerely hope that the majority of the demarcated area can be eradicated soon. But let's see what will be the outcome of the sampling this year.
Okay, thank you. Now we have a question from Manuel Neves from the chat on YouTube. In the surveillance contingency plans, why are the plantations of higher risk only young, lower than 5 years, and old, higher than 15 years for olive trees? What makes us not high risk between 5 and 15 years old?
Okay, so it's a very interesting question. Of course, we need to establish the priority places where the inspectors will perform the inspection. This doesn't mean that we are not doing also the inspection of the olive groves between 5 and 15 years old. Of course, we are doing also these olive groves inspection and sampling. But when we define where we are going to prioritize when the priority places where the inspectors need to go, because it's impossible to inspect all olive groves around the country, it's not possible. We have defined the more risk olive groves, the ones that have now 5 years old, that have been planted with plants before entering into force the obligation to test olive trees, because now it's obligatory before movement, before the nursery be able to market their olive trees, they need to sampling and test for Xylella fastidiosa. This is starting for 3 years ago, I think. So before that, there was no legal obligation to test olive trees. So that's why we select the olive trees till 5 years, because this one, though there are the olive groves that perhaps their plants did not been subject to these obligatory tests before issuing the plant passport, so there is a risk. And also the older ones, because as Clara mentioned, we have a lot of insects in the country, and we need to check if there is cross-contamination or contamination from produced by the insects. But of course, the olive groves between those ages, scales of ages, of course, they are also inspected. But that's the reason we have decided to give priority for these specific kind of olive groves.
But I also can enforce something that, all the old orchards, many times they are abandoned. And that situation makes the opportunity, the vector to go there. When you have very professional olive groves, which are sprayed against other insects on olives, they will take down the Phyllanus pomarius as well. But those old orchards which are more not so abandoned. So that means the opportunity for the better is higher.
Okay, thank you. And the last question is from Francisco Teixeira. That is asking where the Xylella ST's information is available in Portugal.
So sorry, ST, the subspecies and ST.
Yes, the subspecies.
We have all of this information in our website. We have a specific place in our website for Xylella fastidiosa, and then there you can find the despachos and all the information regarding each demarcated area, if we know, because sometimes we don't have the subspecies identification or the ST identification, but when we have it there.
Okay, thank you.
Mainly,
I think
at the species level, it is mandatory to have it because restrictions are applied to the subspecies of the bacteria. There is no difference between ST1 or ST2. What is important is to know it is Xylella fastidiosa, and the restrictions apply to fastidiosa and not to the ST specifically. So the main concern for our information is related to the subspecies.
Of course, ST is important for us in order to try to understand better the origin of the subspecies that we are dealing. But according to the legislation, only the subspecies are regulated, not the ST. So we can have multiplex ST7, ST6, but it's not important regarding the regulatory framework. But for us, it's important to have this information.
Okay, thank you. There's no more questions. So we are going to pass to the last speaker of this webinar, that is Joana Costa. Joana Costa is currently a research and guest lecturer of the Center for Functional Ecology, Department of Life Science, University of Coimbra. Joana Costa research interests are focused on on the detection and research into plant pathology. In parallel, she studies the soil, plant microbiota, and ecosystem service providers, fundamental to the service, restoration, and conservation of agrosystems. For the past year, Joana Costa coordinates several contracts related to detection and regulation organisms, namely Xylella fastidiosa and plant and soil microbiome characterization. Joana Costa is also the Director Technical Management of PhytoLab, Phytopathology Laboratory from the Instituto Pedro Nunes, designated as the official laboratory for the detection of quarantine pests by the Director General of Food Veterinary. And today she will tell us about the current tools for the detection and monitoring of Xylella fastidiosa. Joana, you can share.
Yeah, let me just share my screen. Okay. Okay. So I would like to start by acknowledging the IPB for this. Let me just start again. I have several. Okay, no. I have several monitors and I was not on the right one. So I would like to acknowledge Instituto Politécnico de Bragança for this really important event, namely José Pereira for the invitation, and also to the Ibero-Chifres network, in which I also collaborate, which is really important and allow us to do all these kind of network and dissemination activities. So I will bring you some information regarding current tools for detection and monitoring of Xylella fastidiosa. This has been a long afternoon, but I hope you can follow me. This will be a really detailed description of these methods because in the end, all comes to our results and all the measures are applied given our results. So it is really important for all to understand how we can get to a really validated result. So just a brief description of my institute. Besides being a researcher at the University of Coimbra, I'm a director of Fitolab in Instituto Pedrones, which is a non-profit organization that has been created by the University of Coimbra. It has several pillars of activity. The most known is for the incubation services, but we also have one related research and technological development in which we have 6 laboratories in one, and one of which is Fitolab, Laboratory for Phytopathology. What we do in Fitolab, as Isabel told, we provide specialized services, mainly inefficient controls for plant health. We are designated by the CAV as an official laboratory for some quarantine organisms, and we are accredited by the Portuguese, by IPAC, by the Instituto Português de Acreditação. We have 2 main areas of activity: phytopathology and total diversity of microorganism studies, the currently known microbiota. This is our team. We are currently hosting and contracting 8 researchers permanently in our lab. Some of them have, besides being technicians, they also accumulate with our functions. Most of them related with accreditation process. Sara Rodrigues is our quality advisor. Eva Garcia is the responsible for the laboratory technicians. Beatriz is responsible for measurement technicians. And we have 2 directors in the Fitolab. And we are focusing 4 areas: microbiology, bacteriology, mycology, virology, and nematology. We also have a lot of students with us. We focus in 3 pillars: services and scientific advice, knowledge transfer, and research. One of our main essays, the one that currently occupies most of the year, is the detection of Xylella fastidiosa, and this is the one that I'm going to devote more attention. We, in parallel, do a lot of studies for some companies related with detection and the determination of the microbiota present in soil, in plants, in waters, in plants, in industry. So this is another area of services. We also have a huge collaboration with several universities and polytechnical institutes, and we received a lot of students to make their professional or research theses. We also have professional scholars, and we receive a lot of schools. We have currently 3 projects ongoing. One of it is Chill Out, which I already coordinated. It's funded by FCT, and these are our past projects and networks, and one of the currently ongoing networks is Iberachifas. So what brings us here is the importance of diagnosis, in this case applied to Xylella fastidiosa. I think we all know that pest and disease play a role in defining the structure and the composition of agroforest systems, and can have a profound effect on these systems, and mostly on the ecosystem service that they provide. So the threat of invasive plants pathogens, it's really important and is increasing, given as a result of globalization, increasing human mobility, climate change, and so on and so on. So this emerging or re-emerging of pathogen results in significant economic and biodiversity losses. So this is a huge area that we have challenging with new challenges every day. In combination with this increase in introductions, we have a reduction in the number of active substances that we can use to control this disease. So if in the middle 20s, we had approximately 900 substances, nowadays we have less than 250. So this increases even more the problem. This food security is especially threatened resource poor countries. So the most devastating problems is not in Europe, it's in other areas of the globe, but in Portugal and in the European Union, we have several different and several emerging pathogens that are cause of concern as an apologist focus, one of which is Xylella fastidiosa. So this diagnosis and the pathogen detection is really important, is central nowadays for determining the preventative and management measures to implement, and it's now integrating what we call integrated pest management. So currently, at least in the European Union region, governments are responsible for protecting agricultural natural systems and to prevent that organisms that are in our country avoid them to export to other countries. Although the private sector has a responsibility to provide clean products, meaning nursery and plants for planting, the protection is considered public good, so it's a responsibility of governments. The concept of phytosanitary protection arose in the early 20th century, in the middle 20th century, and is currently indispensable conservation tool. And nevertheless, all the efforts that have been done in this area, harmful organisms and their products are still responsible for at least 30% of losses in production and post-harvest. So it is a really cause of concern. What is diagnosis? What is detection? This is really important. So diagnosis of a disease plant is determining the cause of the disease of the syndrome on that plant population, while detection is identifying the microorganism or the organism that is affecting that plant, particularly maybe a substrate, and it includes plant tissue, soils, and water. So detection, monitoring, and diagnosis are key components for protecting plant system for invasive pathogens, and the identification of pathogens is the first step in disease management. So we can only manage what we understand and what we know. This is a global demand, so we can see that there are no borders for these invasive pests. So international conventions limit the introduction and spread of plant pathogens, and the most known and globally present is the International Plant Protection, IPCC, through the FAO organization. And currently 107 countries subscribe this convention, okay? And provides information of the status of certain plant pests and tries to describe standards for diagnosis. At the national level, there are what we call National Plant Protection Organization that are responsible for this plant security at the national level. In the Europe and in the Mediterranean basins, the prevention and control of plant pests and disease is a task of EPO, what we call European and Mediterranean Plant Protection Organization. And since some years ago, we also have another level, which is the EU, European Union Organization, and their legislation. So we have several layers of organizations, all of them combine a diagnosis network that is really important since this is a really global and challenging problem. There is another challenge. So in parallel, we have more emergency, more introductions. We have more suitable climate for several of these pests. And on the other side, we have a reduction in number of active molecules to use for control these pests. And we have a reduction in the number of people with field experience and plant pathologies. And this is coupled with a general reduction in government support for the agriculture and the inability of private sector to bear the costs of these diagnosis and these management strategies. So it is necessary to find new forms of agricultural diagnostic service, meaning new diagnostic networks. So regarding plant pest diseases, they can be categorized based on the analysis of risk matrix that assess the likelihood of interest of this organism in the EPO region and the likelihood or the impact that this introduction would have in the territory. So given this risk matrix, the EPO region defines what they call a quarantine pest, which is a pest of potential economic importance to the area, and that is not yet present, or if it is present, it is under official control. Okay, this is related. These quarantine pests, Xylella fastidiosa is one of these organisms, and these quarantine pests are divided in two lists. The A1 list, in which the disease or the pest is absent from the ecoregion, meaning it is not been yet detected, or it is an A2 list where this pest is already present in our territory, but is under official control. So Xylella currently is a quarantine pest belonging to the A2 list. In parallel, there is another classification for these harmful organisms that are regulated non-quarantine pests, RNQP. These RNQP are non-quarantine pests, but given their role in the production or their impact in production, they are also under surveillance. So this is an extract that I taken from the EPO site, where they really clearly distinguish between quarantine pests and RNQP. An example for RNQP is Erwinia millover in our territory, meaning the measure for quarantine pests include exclusion, eradication, and containment, and aim to prevent unacceptable economic, environmental, and social impact resulting from their introduction. While for RNQP, they are intended to prevent an unacceptable economic impact on the intended use resulting from the presence, meaning the pest is already present and we aim to limit their dissemination and introduction in other areas. So in this case, RNQP is also being transferred to the EU law currently. So we have these two categories of these pests, both in EPO and in EU. So one of the missions of the EPO and the IPCC is to deliver diagnosis protocols, what we call standards. When we go to the EPO website, we can find a lot of standards on phytosanitary measures, and one of them, which we can find in PM set 7, is the diagnosis standard. So meaning that EPO has been working and has several protocols and works with reference labs and research labs in order to come up with essays that are able to promote, to give accurate results for the detection of some organisms. This is the diagnosis protocol for Xylella fastidiosa. It is available on the EPO website, and is on its 4th version is under revision currently. So this is, it has 2 flowcharts for the diagnosis depending on the matrix, meaning if we are talking about detection in vectors or if we are talking about detection in plant samples. It is a really intricate and complex flowchart. I will clean it up for you to understand clearly what we are doing, because there are a lot of methods that are proposed here and we have to choose some and not all. So we choose from the available methods and we implement in the lab some of them in order to accurately diagnose Xylella. How is this done in terms of official and legislation according to the EU? So the European Union has a European reference lab for bacteriology, and this European reference lab coordinates some activities with national reference labs. So each country has to identify a laboratory to be designated as national reference lab. In Portugal, it is INIAV. The European Union at the member state also, and each member state also has to identify an official entity for in the scope of the phytosanitary measurements. In Portugal, it is the GAV. The previous talk was performed by the two of members of the GAV. The GAV has several roles in this regulation, EU regulation. One of them is to appoint official laboratories, meaning of laboratories that can perform the diagnosis for these regulated pests. This EU regulation that I have here tell us what these official laboratories must comply with in order to be designated by the competent authorities. They have to possess the expertise, equipment, infrastructure, and staff to carry out such tasks to the highest standards, and it should be accredited for the use of these methods according to the international ISO standard 70025 for general requirements. So these are the two main parameters that we have to meet in order to be designated as official laboratory. The accreditation consists of the assessment and recognition of technical competence to carry out specific activities. In order to be accredited, we have to implement quality systems in the laboratories, management systems to provide quality and confidence in our results. And the accreditation activities are subject to EU legislation, and each country appoints an official body to accreditate. In Portugal is IPAC. So we have to be audited not only by the GAV and the European Union, but also by IPAC in order to perform this kind of assays. Since we have such a particularly metrics and context, EPO has translated what these ISO accreditation regulations have into a norm, into a standard that is standard PM 798, where it details how we in plant health authorities should comply with the accreditation ISO 70025. So we have to comply with several obligations. One of which is what kind of methods can we use? We can only use methods that have been validated and are described on standards, meaning EPO standards in our case, for instance. If these methods are not available, meaning when a new pest arrives to the continent or is for the first time detected, we have to develop these methods, and these methods have to go through a very thorough validation process in order to assure all the performance criteria and to assure the quality of the results. So the terminology in our case is use standardized methods that have been already validated and been described in standards. And we have several, we have EPCC, we have EPO, we have several standards that we can use. And we can also use non-standard methods if we can provide validation details for those matters. So what happens is that EPO standards need to be implemented and need to be taken by the European Union, and the European Union has defined within the EPO standard which methods we can use at the European level. So from all of those methods, we can only choose what the European Union has elected and selected as the most, as the ones that they think is the best for our context. So we have to go for the European Union. We have to see what the legislation identifies, and we have to implement them in the laboratory. The terminology in our case is really difficult because there are several synonyms for the same thing. So I think it is important for us to understand that an assay, a method, a test, a protocol, diagnosis protocol are being usually used in the literature as synonyms, okay? For us, a test, and in the EPO region, a test combines three things, always: a method that can be, for in this case, real-time PCR, one target pest, in this case, Xylella, and one matrix, in this case, the bacillus from plant material. So when we are doing the diagnosis for Xylella, we perform at least 3 different tests because we use 3 different methods. If we go for vectors, we use 6 different tests to come up with the results. Okay, so just to understand how complex this is, and for each test we have to have verification performance characteristics. So the EPO diagnosis protocol is a sum of this combination of tests that the European Union, in this case, and our national official entities select which we should use. It is really important to understand that, and depending on the country that we are talking about, the tests that have been implemented can be different because the choice of the test or the combination depends on the circumstance of use. And if rapid detection and accurate diagnosis is essential, depending on the purpose of this diagnosis, we can select different tests. And these are just several kinds of objectives to perform a diagnosis assay. It can be done for routine surveillance. It can be done for surveillance of latent infection. It can be done for surveillance in eradication program, sorry. It can be done by routine diagnosis. So there's a lot of different objectives, and depending on the objective, the choice of the tests and combination can be distinct. Okay, so this is really important. So this is not one size fits all. This is more or less a tailor-made decision depending on the purpose of the assay. So depending on the circumstances, different tests and characteristics are needed. So what is the difference between method selection and verification? This accreditation and these norms, ISO 70025, has changed recently and currently is based on risk analysis. So we need to go into our context and determine which kind of performance test we want to test and which kind of performance criteria we have to evaluate. A validation process is really, really difficult and detailed process that is done in consortium. There are projects like ValiTest, which was a huge project from EPO that gathered a lot of reference labs in order to validate new tests and come up with performance criteria. After this is done, they are published in standards, and the current official laboratories just have to verify if they can use these tests in accordance with the objectives. So these are some criteria that we have to analyze: the sensitivity, the specificity, meaning how much can we detect, what can we detect. Are we just detecting our target pathogen, or we are mixing with and have cross-reaction with others? Can we eliminate the effect in our lab of the operator of the equipment? Can we have a repeatability in our lab, meaning if I test the same sample several times, I will always come up with the same result? And how is the metric variation, the selectivity, impacting my assays? So when we are talking about the selection of a test, we need to know that sensitivity and specificity normally are not together. Meaning, when we increase sensitivity, which is what in the case of screening tests, we are diminishing sensitivity, and the opposite is the same. When we are going for increasing specificity in case of identifications, we increase sensitivity. So we have to be aware that the limits of each of the tests that you use in order to be sure that we give the correct diagnosis. So what usually occurs is we start by doing screening tests with high sensitivity in order to catch even false positives, okay? And then we confirm the result, narrowing down these false positives, increasing the specificity. This is the EPO standard, Xylella, and as we can see here, we have a lot of screening tests that we can use, and then we have a lot of other tests that are related with the identification and the sequence type determination. This is our protocol in Phytolab, and we've selected among the tests that the European Union and GAV allow us to use. We select for the molecular detection 2 tests. We have the processing of the sample, meaning we have to cut the bacillus from all the leaves that we receive. We have to all the heads of the vectors. We have to perform the total DNA extraction. We have to have an internal control to limit the existence of inhibitors in this reaction, and then we perform the molecular detection. This molecular detection is based in 2 methods depicted and described in the EPO standard, Appendix 5 and Appendix 7. For those who are familiar with the Xylella, it's Harper and Young methods. And if, when applicable, we go ahead and confirm this detection by using a conventional PCR method described in the Appendix 4 and Misavange et al. 94. So what we have to do is, depending on the results, if we have 2 positive tests from these molecular tests, we have Xylella detected. If we have both tests negative, Xylella is not detected. If we have some inconclusive results, then we go ahead and perform the PCR. We have, it is important to notice that all of these tests are done in duplicated and with replicates and dilution. So at the end, we have several results that they can use to give a confidence result. What are these molecular detections? So briefly, they are based on primers. We are small pieces of DNA that we use to select specific parts of genome of the bacteria. So the specific primers only amplify Xylella fastidiosa DNA fragments, okay? Which is the difference between a PCR and real-time PCR. The method is the same. The difference is that the PCR, I only see the results after the PCR, and I have to put them on an agar-azo gel and see the amplification in a transilluminator under UV light, while in the real-time PCR, I can see the amplification of the products in real time. So I'm looking at the computer and I'm seeing the amplification accumulating, since the fluorescence will be directly proportional to the amount of the gene that is being fragment that is being amplified. So I can see this kind of result. What is really important is that not only the shape of the curve, meaning it has to be exponential, but also this CQ value. This CQ value is in which cycle, number of cycle, this amplification started. This is really important in our evaluation of the results in order to determine if this is a positive or negative result. So these are the basis of our molecular detection. And the three methods that you use are all based on different parts of the genome of Xylella. So we are sure that we are amplifying Xylella, not cross-reaction. Okay, so this is the sum of our results of our protocol. So we have the molecular detection by 2 methods, and if we have inconsistent results, we go for a third method. Okay. Why? This is for detection. Okay. In some cases, detection is enough, meaning the presence of the absent or the absence of the pathogen is sufficient for the authorities. For instance, when we are issuing a phytosanitary passport, this is detected or not detected. But when we are looking particularly with Xylella, the determination of the subspecies, as Engineer Clara was telling, it's really important because restrictions are applied to the subspecies. As we heard earlier in the previous talks, there are several distribution and several focus of Xylella outbreaks in Europe and worldwide. And what we know is that the introduction in Europe has been done in several events, okay? And each event probably can account for a different subspecies or a different FST in Europe, and why it is so important to know which subspecies it is. Looking at this sum, we know that Xylella has at least 3 subspecies that are really important for us: fastidiosa, multiplex, and pauca. And knowing the subspecies, it directly correlates with the host plant list. So what the measures applied when we have multiplex in terms of eradication and restrictions is totally different from what we have to apply if we have fastidiosa, because the host plant list is different and specific for each subspecies. So when we look at the number of host species, this is the count, and it's a little out of date. The last update, and you can consult it in the European Commission database of host plants, is from June 22. And this update, if we count for detection based on 2 methods, we can have 423 host plants that are susceptible to Xylella fastidiosa, and this is the list for the species. But when we go to the legislation, we see that there are narrow lists for the subspecies. So if we can come up with the subspecies, we can narrow the list of host plants that can be subject to restrictions. And how can we account for the subspecies? We can use, and this is a method that is now being integrated in the EPO standard with all the validation data that is necessary, and is also based on a real-time PCR, and it is used to determine the host range of the Xylella fastidiosa subspecies. So as we can see here in this result, this is what we can get from the real-time PCR, and each color here results from a set of specific primer for each subspecies, and it's related with distinct subspecies. So if I have one curve in this PCR reaction, meaning I have a specific fluorophore for this reaction, I have a multiplex. If I have a result with this fluorophore, I will have a fastidiosa, and so on and so on. So this is a really important and really important way to determine subspecies. This a real-time PCR is even more important when in areas where we don't have the isolation of bacteria. As probably you know now currently, the name fastidiosa from Xylella comes from the fact that it's really difficult to plate and grow this bacteria and isolate it. So all the methods that we come up that avoid isolation are really important in accelerating the process of diagnosis. So if determining the subspecies is important for the host range, when we talk about epidemiology, meaning who is here, where it has come from, how it is connected between outbreaks of Xylella, we need to go for a fingerprinting analysis. And this is done by multi-log sequence type. And this multi-log sequence type enables us to determine the sequence type of this specific isolate of this specific bacteria. MLST is a really well standard method that was developed for human pathogens, and it has been now for several years developed for Xylella fastidiosa, and it is based not on the whole genome but on the sequence of 7 housekeeping genes. Housekeeping genes are genes that belong to the core genome of the bacteria. These are common to all subspecies, so they are conserved among the species Xylella fastidiosa, and they are housekeeping because they are under purifying or neutral selection, meaning they evolve slowly during time. They only evolve by accumulating point mutations. So what we do, and this is a really demanding and time-consuming process. If we have the isolate, is really nice because we can extract DNA from the isolate. Otherwise, we have to use total DNA, which is much more difficult. We have to amplify 7 different genes by PCR. We have to purify these genes and we have to sequence them. So we have to determine the sequence of these genes, and then we have to curate the results and submit this sequence to a platform that has been developed for Xylella, which analyzes them. So the process is we do the PCR, we obtain the sequences, we pick up each sequence from each allele and we submit it to this platform, and it retrieves us a number. This number for each allele may be a number that has already been described in other parts, so it is a known allele, or we can even have a new allele because we are seeing some diversity within Xylella fastidiosa. And the combination of these 7 alleles give us the sequence type for the bacteria. And this sequence type is really important when we are talking about epidemiology, because it is the only way that we can determine if the sequence type has been identified in several other parts of the globe. If it is the same that has been previously determined, it's the only way we can do epidemiology and trace the infection route for these bacteria. So just to finish, this is the kind of works that we can do with these results. These are some isolates, and we can see here the allele, and this is the number of the allele, and we can determine how closely related the isolates are, and if they are new introduction, if they are related to other parts, and so on and so on. So we have 3 levels of assays. We have the diagnostic, we have the host range determination, and we have the epidemiology. So this is the kind of results that we can obtain from determining the sequence types. Focusing on the host, we can see which sequence types have been isolated from this host. So we know, for instance, for grapevine that only fastidiosa has been determined, has been detected in grapevines with all of these ST. We know that ST1 is able to infect grapevine, olives, almonds, citrus. So all of this kind of epidemiology we can do based on this information. And this is the result from an assay done in Mallorca, and you can see that there are several subspecies detected, and different sequence time between the islands. So in Ibiza, we only have subspecies Pauca, while in Majorca and Minorca, we have subspecies multiplex with same sequence type, but in Majorca, we have two different sequence type and subspecies. So this can give us some feedback about the introduction and epidemiology of the disease. So I will skip these next slides because otherwise you will be, and I will give just a feedback on where you can find all the information related with the diagnostic expertise. It is really important in networking and when we are starting doing this kind of works. EPO has a database for diagnostic expertise. It is up to date. Every year we have to validate information. You can find the laboratory lists and you can search it for organism, you can search it for country. It has also an expertise list, meaning you can find researchers or technicians depending on the expertise that you want to implement or if you have doubts. So all of this information is made available in this database and is really important. I would like just to end by inviting all of you, if you are in Portugal, if you want to join us in an international workshop that we are going to hold in Fundão. We are going to keep discussing this emerging disease in horticulture in Portugal, namely Xylella. We'll have some international researchers devoted to Xylella. So if you want to join us, it will be on the 5th of May. And just to thank you all.
Thank you, Joanna. I have a question, and there's also questions from the chat from YouTube. My question is, these tests are expensive and time-consuming, right?
Yes.
So, do you know if the scientific community are developing detection methods more faster and more cheaper?
That's a really good question. As I mentioned, we have to be aware that sensitivity and accuracy are really important, and depending on the status of the infection, some other more affordable methods can be implemented. There are some ELISA, there are some even tests that can be on the field, but it is also a compromise between accuracy of detection and where we are now. So we are talking about a really devastating organism, a quarantine pest, and until now, these are the methods, the more affordable methods that we have. I didn't go into detail in how difficult it is to do the sample collection. We have to cut all the bacills from more than 100 leaves for each sample. So this is really time consuming in terms of human resources. It has to be really trained human resources in order to identify symptoms, in order to know. We have more than 100 hosts, so they have to know which part of the plant they have to take. So all of these are really demanding in terms of human power, and all the methods are based on molecular methods that are really expensive. So there are other methods that are more cheap, but there's a compromise between specificity and accuracy of results.
Okay, thank you. So now I have a question from Manuel Neves. If the detection, how flexible are the primers? Are they available or change according to the subspecies or sequences type? These determinations have to be done separately?
Yeah, so the primers are different, meaning all the primers are available in the EPO standard. Everyone can just order them. You just need to have the authorization to work with Xylella from the GAV. The tests are made separately, depending how are you determining the subspecies and the sequence type. Meaning, if you have the sequence type, you obviously have the subspecies. Okay, so you can use the combination of 2 of these 7 genes to determine subspecies. This is described in EPO standard in, I think, appendix 14. So you can just amplify 2 of these genes and you can have the subspecies, okay? And if you want the sequence type, you just sequence the other 5. The other way, which is much easier and in terms of time consuming and accuracy of the results, is doing a real-time PCR. And this real-time PCR, the primers are not the same as the one for sequence type detection.
Okay, thank you. I have one last question. If agricultor or producer suspects that one plant is infected with Xylella, what's the steps to follow?
I think Engineer Clara can answer that for me.
Yes.
She's here. If she won't mind.
Yeah, can you listen? Yeah.
Thank
The
you.
question is if when you are speaking about quarantine disease or a pest or can be an insect or disease, if any lab which is detecting that, even it is an authorized or not authorized lab, if it is a positive finding of a quarantine disease, immediately have to notify the authority, let's say the GAV of that finding. I don't know if Paula wants to say something about that, but it's even when you send a sample to a member state and in those lab in the member state you find a positive, there is a network that that member state will notify immediately the Portuguese authorities of that finding. That is the procedure of the, because and even by our legislation, anyone that have a knowledge of to be or suspect to have a quarantine pest should notify the official services. That is the obligation. But the labs are obligated to that automatically.
But the producer can contact the DRAP, the GAV, one of the authorized laboratories in order to ask for details on how to collect sample, because the sample collection is really important and they can determine the fate of the detection. So all these procedures should be taken in account and should contact the authorities in order to understand which kind of samples they should collect, how should they send them to the lab, noting that if the result is positive, as Engineer Clara was saying, we have to communicate it to the national authorities, okay?
The question here, when it is a sample taken by a private person, which is not an official sampling, and therefore, if we are in the presence of someone took a sample from his garden and sent to the lab and it becomes positive, I think we have to see if, because that sample, it was not official taken. And in that circumstances, we have to have an official sampling, not by a private person, because then you are dealing with a difficult situation. Because, for example, if I decide to take a sample in my neighbor and it is a positive, and then I go to say my neighbor have that problem, how trustful is that type of sampling? Yeah. Sampling, it has a value if it is collected by an official. Okay, if you suspect that you have a sample, a positive plant in your garden, the best way is to apply for the regional services, for the inspectors to take a sample, not to do it yourself. I think you should do it. The person should take care to have an official sampling, and that official sampling it is taken according to the rules. But I can underline this situation. If it is a private sample taken by someone that, for instance, it is taken a good sample and it is positive, it makes a kind of situation that you have to have an official sampling. Otherwise, you can be in a situation of what is that? What is that positive?
Okay, thank you for your answer. I don't have any answer, any questions, and also in the chat there's no questions. So I would like to thank all the first speakers from today. And in total, we had 7 speakers in this webinar. We are at the end of the webinar. For the participants enrolled in in-person webinar, we see you on Thursday. Hope to see you soon. I don't know if Professor Alberto wants to say some words. He's telling me no. So thank you so much, and it's the end of the webinar.
Thank
Thank
you.
you.
Thank you.
Bye.
Bye-bye.
Bye-bye.
Bye.
Thank you. Goodbye.
Bye.